So thank you very much for having me today, and we will be talking a little bit about head and neck cancer. So I am translational cancer scientist. So the first question is, what is translational cancer research? And it's relatively new branch of science, and the purpose of that is to move discoveries from the laboratory to the patient's bed as quickly as we can. So that's exactly what we do. In our lab, we focus on the projects that we think have the potential within X amount of years, from 3 to 5, to reach the clinical trial in real patients. So why it is even important to study head and neck cancer? First of all, the numbers of head and neck cancer are rising all the time. Right now, every year, approximately 900,000 patients are newly diagnosed with head and neck cancer. And unfortunately, many of the patients die because we don't know yet how to treat those cancers well. Even those patients that are treated and cured, the effects of the therapy can be long-standing. Surgery can affect the ability to breathe, to talk, to swallow. Right, so we want to find the ways how to treat the patients with head and neck cancer in ways that will first cure them, and second, will strip them from the side effects that the therapy that we're using now can actually cause. So what is cisplatin? Cisplatin is the chemotherapy. It's a drug that approximately 60 to 70% of patients with head and neck cancer will get. Or cisplatin or another drug from the same family. Now, it's a good drug. It's been used for many, many, many years. The problem with this drug that when it's given systemically, it's given to the patient, to the vein, right? It's not just affects the cancer, it affects all the cells that divide quickly. It doesn't go and find the cancer cells and just kill specifically them. No, it kills all the cells that grow quickly all the time. That's why the patients that are treated with this drug systemically, they have very bad side effects. So we need to find a way to deliver the drug better. So we're working on head and neck cancer, and in head and neck cancer, some of the cancers, not all, or not all of them, but some, we can see because they're in the mouth. So we can ask the patient to open the mouth, and we can see the tumor right there. So one would think that why to give the drug systemically? Why not to inject it in the tumor itself? It seems like a good idea. No, if you take a free drug, not anchored to anything, and you inject it in the tumor, first of all, you have to inject in multiple spots across the tumor, because if you do it in one spot, the drug will stay there. Second, when you just take a free drug, inject it in the tumor, it will dilute out of the tumor quickly and get into the blood vessel once again. So that's exactly why the intratumoral injected of the drug is not a great idea. And there were several trials across the last 15 years that attempted to do that, but it didn't work well. So what we thought that we need to do something better. We need to create a system that keeps the drug in the tumor without letting the drug to get out of the tumor into the blood system and affect other tissues besides the tumor. So what we created a nanoparticles, that's small circles, conjugated, bound to the drug. Now, there's something special about those particles. We created them as a patch. So instead of injecting the particles into the tongue, we created a sticker. Why the sticker is better? Because the sticker covers a larger field of the tumor. So you can, if you see the tumor, you can take a sticker, put it on top of the tumor, and it will cover the entire tumor instead of injecting many, many, many times. And there's something special about this patch. So when the nanoparticles with the drug, when they get into the tumor, when they touch water, they get bigger and they sit in the tumor for a long time, and the drug works just in the tumor exactly where we need it to be. The first thing that we did, we tested it in mouse and we tested in hamster. Why do you think we tested in hamster? Why mouse was not enough? Hamsters that have something that mice don't have. They have cheek pouch. Everybody saw hamsters stuffing nuts and food into their cheeks. So this is a place where we can grow a human-sized tumor there and then apply the patch on top of the tumors. So what we did here, we took a hamster cancer cells, injected them into the cheek pouch, let them grow to the size about 1.5 centimeters, and then we took those patch, the patch with the drug, applied it on top of the tumor. And this is how it looks like on the left on the left side. What you can see here in the middle, it's a blank page. It's a page with nanoparticles, but without a drug. And on the right side, it's a PRV111. That's the name of the page. That's the page with the nanoparticles and with the drug. I know it's hard to see, but the hamster that was treated with a blank page, it has a very big and nasty tumor there. But the hamster that was treated with nanoparticles with a drug, the tumor almost completely gone. After doing a lot of work after that, we applied this page in a very phase 1, it's an early trial, to 8 patients. Those were the patients that they had a scheduled resection of the tumor. So they had the surgery planned. And we had a window of 4 weeks to apply this page before the surgery anyway. And as you can see here, the chart here shows decreased size of the tumor after the patches. And out of the 8 patients, about 5 had complete response. The tumor completely gone. When they went to surgery and the surgeon cut the tumor and the pathologist looked at the lesion, there was no tumor cells left. So right now it's in the phase 3 trial, and we're waiting for the results, and hopefully it will get approved and we'll actually make it to the patient. The second story that I want to talk about is detection of the cancer. So in head and neck cancer, if the cancer cut in early stage, stage 1, almost every patient can be cured. Almost every patient, like 80 to 100% of the patients can be cured. When it's detected at late stage, the number drops down significantly to about 25 to 40%, depends on the stage. And the problem is that many tumors detected in dental offices, because dentists, they're the first one who sees those things. Now, the problem is that many dentists, they're not trained well enough to distinguish between the cancerous lesions and non-cancerous lesions. As you can see here, the top and the bottom pictures, they all look the same, just a white sore, right? But one of them is cancerous, the bottom one. But dentists, many of them, they don't know how to distinguish between the two. As a result of that, 82% of the patients having the biopsy, the painful resection of the lesion, just to learn that it's not cancerous. At the same time, the patients who need the biopsy because they have the tumor, they may not have it on time because the dentist may say, yeah, no, just let's wait for another couple of weeks and see if it heals. So we wanted to develop a tool to triage the patients and to know exactly which one needs a biopsy, which one is not. So what is liquid biopsy? Liquid biopsy is detection of something that coming from the cancer in body fluids, in saliva, urine, blood. Cancer, when it grows, it sheds the tumor cells and material from the tumors into body fluids. And sometimes we can detect it and learn something about the cancer, make it informative for us. Now, why liquid biopsy may be better than a traditional biopsy, which is just resecting, cutting a piece of the tissue? First of all, it's not painful, right? If you're collecting saliva or the blood, it's much less painful than cutting the tumor. But the best advance is that you can repeat liquid biopsies over time. You cannot cut the biopsy every time from the patients every week or every 2 weeks, every month. With liquid biopsies, you can do it every week, every month, if you want to, every day. So what we did, a long time ago, our group showed that cancer of an oral cavity shed cancer cells into saliva. So we thought, why not to use saliva to triage the lesions? Saliva, it's even better than the blood because everybody knows how to spit. For the blood, you still have to go to the hospital. You have to have somebody to take the blood from you. Saliva, you can collect in the comfort of your own home. Right? We can send the kits to you, you spit, you send it back to us, and we can run analysis and do that. So what we did in 2022, we collected all available genomic data on oral cancer. So every single cancer that we can put our hands on that was sequenced at the time, it means that we know the genomic composition of those cancers. We pulled them together and we asked a question: can we create a panel of small number of genes that when they mutated, we can distinguish between cancers and non-cancers? So this is exactly what we did here. We collected all the cancers that we had access to at that time, and we created a panel of 7 genes that are labeled here. And in this panel, approximately 85 to 90% of the patients have at least one mutation in one of the 7 genes, right, in 90% of the cancer patients. So what we did then, we created a special kit that only sequences, only assesses presence of the mutations in those 7 genes. And then we applied it to 121 patients, freshly diagnosed, before they have a resection. So they come to us, donate the saliva, they spit in a special tube, and then they go to the surgeon. So at that point, we don't know if they have the cancer or they don't have the cancer. Now, all of them, all 121, eventually had cancer. But what we did, we applied our kit on the saliva from the patients and on the tumors resected from them. And what I want to show you here, that at the left side, that's the most mutated gene in head and neck cancer. It's called P53. The line here, it's a schematic representation of the gene. And the lollipops are location of the mutations in all 121 patients in this one gene. What you need to see is that the upper line is the distribution of the mutation in this gene in the primary tumors. At the bottom line, in the saliva. And you can see they're pretty much identical. So all the mutations that we found in primary, in any tumors resected from the patients, we can detect the same mutations in their saliva. And altogether, 93% of that we see in the tumors, we can identify in the saliva. We did lots of work after that. We established another company called DX. Right now, we're in the middle of the clinical trial with Tempus, and that's how the kit looks. That's the box. And we hope that will be available for the patients, hopefully by the summer. The final story that I will share with you is discovery of the new drugs. So we talk about how to deliver the drug to patients, how to diagnose the cancer, now how to detect the new drugs, how to find the new drugs. So head and neck cancer is hard to treat. That's why we don't have new therapies for about last 15 years. They're extremely heterogeneous. It means that they do not look alike. 2 patients that have the same tumor, they're very different genetically. The patient that may respond super well to that drug, they will not respond to the second drug. And yes, all those bad things that makes it very difficult to treat head and neck cancer. Most of the drugs developed up until now, they are specifically targeting just one protein. So many cancers are driven by proteins that not functioning well. And when you inhibit only one gene or one of the proteins, the tumor knows how to compensate. You will inhibit or stop the functioning on one protein that we think causes the cancer. But over time, the tumor may switch and use other proteins to do the same thing and not to die. The hypothesis is that instead of focusing on a single protein, you have to focus on adapter proteins. Adapter proteins, they're proteins that you can think about them as a hub, as an airport. What they do, they connect the planes, the many proteins bind to them, and then they send the signals downstream that tell the cells not to die, keep growing, or die, and don't keep growing. So by inhibiting adapter protein, by cutting the hub, you cut in many signals together that lead to cancer instead of just one. So we collaborated with a company in Silicon Medicine. That's a company that we have established a long time ago when I was in Hopkins. We uploaded all genomic data from 1,000 of head and neck cancer patients. And we asked the system to find the adapter proteins that the system thinks are the most possible target for the drug development. And the top ranked protein was PDC-10. And after we found that, we looked at the patient's data. So on the left side, you see the chart that shows the patient survival. The red curve shows the patients which have very high level of this adapter protein, very high. And the blue curve shows the patients with low expression level of this gene. As you can see, in patient tumors, the patients that have the higher level of this gene, they have a worse survival. Then when we completely deleted this gene in the mouse. We induced the cancer in a mouse, and at the same time, we deleted this gene completely. And as you can see here on the right side, the black curve, that's the mice with a normal high PDC-10, and the tumor grows pretty fast. But when you completely delete, when you eliminate the tumor from this gene from the cancer slows down. So that made us think that we found the target, and we potentially can inhibit this hub and potentially treat the patients. So how do you inhibit adaptor protein? And again, like think about adaptor protein as an airport that have a couple of terminals. You don't want to inhibit the all airport. You just want to cut a terminal that leads to cancer, but at the same time, you would keep the functional, all the terminals that do not lead to cancer. So how do you do that? You take a small peptide, which is another protein, but very tiny, and it binds to a very specific location on top of adaptor protein and blocks just one hub, just one terminal that leads to cancer. We'll keep it all the rest functional. So that's exactly what we did. We designed a peptide like this, and we tested this peptide in mice. When you take the cancer cells that have high expression of PD-10, of this hub, and you add our peptide to those cells, it pretty much kills the cells. The cells die pretty quickly. When you take the same cells and you delete PD-10 from those cells, and then you put the peptide, it does nothing. Why? It does nothing because it's nothing to inhibit. That shows that the peptide is very selective and it only kills the cell that express high level of PD-10, like the patient's tumors, but at the same time, it will spare the cells that do not express PD-10 at the high level. Unlike other two stories, this is not yet in clinical trial. But I think that if everything goes well, within 2 to 3 years, quite possible that we will start a phase 1 trial, which is an early trial, just to see how it works. We probably will start it in about 2 to 3 years if all that we plan to do will actually work. And with that, we will stop here. And Nishan Tagrawal, chair of the head and neck surgery, Li Shen, and Dr. Rosenberg, Pearson, Dr. Volks. I'm not a clinician, I'm a scientist, but I'm doing the preclinical work, and I moved these things to the trials. Without those people, anything that I do would not be possible.